Good morning, everyone. So I'm Chi Liu. I'm from Vanderbilt University Medical Center. So it's my great honor to present my work today. So my work is about adaptive nearly label graph optimized for single cell and spatial clustering. So a little bit of background. So the single cell clustering is very popular right now. So the method is generally adapted for the traditional clustering method, such as k-means, hierarchy clustering, community detection methods. So among them, the community detection methods is very popular. So this is a toy example showing how the community detection method work. The first step is to build a k-nearest label graph. So here, k is a hyperparameter, is a universal and fixed k for all the cells. So generally, k will be set to 20 or 30 by default. So for example, we can see here, if we set k equals 10, then there will be, then we'll build the 10-years label graph. To make the graph more robust to outlier and the noise, so the second step will be build a shared nearest label graph. Then the cluster will be identified, trying to maximize the connection within the cluster and minimize the connection between the cluster. So these methods are very popular and very successful in detection the clusters, but they all face the challenge to detect the real cell types. So there are several methods, specially tailored for real cell type identification. I list 3 here. The file is, to detect the real cells based on they will assign a rarely score for every cell based on the neighborhood density. So Gini cluster is the one, trying to identify abundant and the rare cells simultaneously. So the first thing, they're trying to select the informative genes. So for the rare cells, they use the Gini index. For the major, the abundant cell types, they use the final factor. Then they kind of combine these two, coming up with a cell clusters containing both abundant and rare. So gap cluster is a method also based on the k-nearest label graph, but it's based on the local distance change. Here, you can see here it's a very brief example here. You can see the red one is a real cell cluster containing 3 cells. So if you build a k-nearest label for graph, for this one, you can find the 2 nearest label. Then you want to go to the 3rd nearest label, there will be a dramatic distance change, from D1, D2 to D3. So they used the second order derivative of this distance to find those dramatic distance change. Then use a skewness to identify those real clusters. So these methods, in the real practice, we found, so for example, this one, they can both identify the abundant and real, but we found it generally will sacrifice the performance on the abundant cells, trying to detect some real cells. So I will show you some example in the real case. So the gap cluster only focuses on the real cluster. The file is focused on the real cell. They don't identify cluster at all. They're also not very good at detecting the real cells, as they'll show you in the real application. So the major challenge here is, how to identify those real cells accurately. And the second challenge is how to identify the real and abundant simultaneously and accurately. So in the daily RNA-seq, single-cell RNA-seq analysis, sometimes we found, we actually saw a group of cells, in the UMAP, very separate from other cell clusters. But no matter how high resolution we go, we cannot separate those cells, from the major cluster. So we found, the problem might be those fixed and the universal K for all cells. I also use this toy example to show you. So here you can clearly see 2 cell clusters there. So one is relatively minor, one, the other is more abundant. So if we set K, for example, we set K equals 10, then that means, for this cell, we not only collect 5, we find the 5 nearest labels. Then because the K equals 10, so we will force this, cell connect to the major population there. These 5 are all false connections. So that means you will have a lot of false connections between the real abundant cell clusters. That means, then it's hard to separate this real cluster from the abundant ones. So then, when people will ask, so why just use a small k? Sometimes a small k, probably good for this real cluster, but for this abundant one, if we use small k, that means we focus on the local distance. Then that means will lead to the over clustering of these abundant cell types. So we found the solution will be, how about we can choose k adaptively for each cell. Then for those real clusters, we have a small k. For those abundant cells, we have a large k, so that's everybody fit. So that's basically our idea here. So our idea is, I actually borrowed the idea from this paper, Neural Computing, from 2022. So this is a kind of a pipeline here. We have 5 steps. So here, the first step is still build the k-nearest label graph. The k is fixed and universal. So then you will have, for example, k equals 10, you have 10 nearest label graph constructed. We use 2 cell example to see how the k is adaptively changed for each cell. So we will set, this kind of, we will set a cutoff. Cutoff is a function based on the 10 distance, from the k-nearest label, 10 nearest label. So it's a function of the 10 distance sort of from the lowest distance to furthest distance. And the hyperparameter delta. Delta is kind of, the sensitivity is a parameter, how sensitive to the local distance change. So this cutoff, so we select the k if the dk less than d cutoff. So this kind of cutoff is borrowed idea from this paper I mentioned. So for example here, the cell A, you have a 10 distance. Because they only have 5 nearest label here, so from D6, they will have a dramatic distance change there. Then the D cutoff will be set at this location. So only the D5 less than D cutoff. Then the cell A, then the nearest label will be set to 5. For this cell B, because they come from a abundant cell type, they will slowly increase from D1 to D10. Then D cutoff will be set here. So that means, D10 is still less than D cutoff. That means the K will be set to 10. So that means in this way, if we choose K adaptively for each cell, that we will remove those phony connections between the real cluster, the abundant cluster. We also build the shared nearest label to remove the potential outlier noise. Then we can do the common, you can use any community detection methods. Then we add optimization steps to automatically choose this delta parameter here. So because, we want to get the most of the adaptive strategy and not too sensitive to those random noise. So I will show you several examples. One simulation, kind of 3 real, but we have a multiple kind of data sets in the paper and also in the GitHub. All the manual there. So this is a simulation data sets. We have 2 settings here. Every setting, have 1 real cell types and 2 abundant cell types. So the setting 1 is more challenging than setting 2 is because in the setting 1, the real cells are more similar to one abundant cell type. So you can see here the red is the adaptive nearest label. The blue is a regular k-nearest label. The different shape is different resolution. The axis is, how many real cells in the setting. So we start from the, only have 2 real cells, 220. So the y-axis is the F1 score. So you can see here the red one, the adaptive k-nearest label always retrieve the perfect kind of F1 score. But for the blue one, the regular k-nearest label, when the real cells are very few, it cannot detect at all. It cannot separate from the major cluster. So, and also, at a certain point, it's depending on the resolution. For example, when there are 14 real cell types here, you go high resolution, you are be able to find those real cell clusters. But if this high resolution is low resolution, it cannot be found. So this is kind of an example for the one simulation. So this is a reference. That means, you know, the standard. We have 2 abundant cell cluster. We have 1 rare, have 10 cells there. So for the adaptive K-news label, we can clearly separate these 3. For the regular K-news label, you know, we cannot separate this cluster from this red one. You know, it's belong to the red one. If we go extremely high resolution, you know, so the abundant, you can see here, the abundant cell cluster actually have the overclustering problem there. Even at this stage, even you overcluster the abundant cell types, you can still distinguish the rare cluster from the abundant cluster. So let me show you several real applications. So this is a kind of a simple data set from the mouse intensity published in 2017. So this is for the adaptive K-news label. This is a regular K-news label. Then have the same resolution. This is a K-news label with very high resolution. So the adaptive K-news label, you know, find 10 clusters there. So each cluster have very well-known gene markers marking specific cell type. Especially, you know, for example, you know, the ninth cluster, from 0 to 9, the 9 cluster only have 3 cells. Actually, the panel cells, because they have specifically those deficient genes expressed. The A cell is tough cell. They have the HiTseq, LRMP, those marking tough cell markers. So for the K-news label, you can see they cannot detect those panel cell and the tough cell. They are kind of, you know, the panel cell is misclassified as a goblet cell. The tough cell is misclassified as one of the enterocytes. So for especially, you know, so even you go very high resolution, you cannot distinguish those panel cell from the goblet cell. You cannot distinguish the tough cell from those enterocytes there. So this is, you know, how those real cell cluster specifically tailored for real cells detection methods. So the gap cluster, for example here, you know, the gene cluster. The gene cluster here, you can see, you know, the gene cluster is the only one can identify the abundant and real cluster simultaneous. You can see they even not do a good job at the abundant cell cluster identification. They cannot separate these 2. They cannot separate all kinds of enterocytes. For example, stem cell, the transit amplifying cell, and those intermediate and the mature enterocytes. They cannot, you know, they also didn't do a good job at real cluster identification. They cannot identify the panel cell. They cannot identify the tough cell. So the gap cluster, you know, they identified the tough cell and the panel cell, but they also identified the relative abundant, the goblet cell, because the goblet cell have almost 100 cells there. They identified the rare, but missed, for example, those K cells and enteroendocrine cells. The file is, you know, it do a miserable job there, so they cannot identify those panels and the tough cell. This is how show you, this is the right panel show how the delta, how we automatically choose the delta, optimize the delta value. So we find the delta, you know, before it has an increasing number of clusters or increasing number of singletons detected. So here, for example, at the -0.9, there will be a dramatic increase of a number of clusters and dramatic increase of singleton. That means, you know, probably you have a random noise coming up. So we set the optimal delta automatically to -0.9. So this is the default delta. This is the optimized delta. We can see the difference here. We can find those, you know, there are 2 types of enterocytes. So one is stain, one is transit amplifying. So for the optimal delta, we can find the panel cell. So this is a more complicated, example from the human pancreas. Human pancreas have a lot of, cell types, and endocrine cell types, and fibroblast, stroma cells, and also immune cells. I will not go into detail because it's too complicated. Let me just bring your attention to this cluster. So the first figure is the manual annotation from the paper, from the original paper. I want to bring your attention to this population. This actually very close to each other. Actually, is three immune cell types. One is T cell, one is mast, one is macrophage. So for the adaptive kinase label, you can see they can clearly separate these three out. But for the kinase label and the kinase label at a very extremely high resolution, they only can find one cluster there. They cannot distinguish these three. And also you can see, compared to the manual annotation, we actually find in this cluster, we actually find the two small cluster, separate two small cluster out. So let me bring your attention to the 21 we found is a duplex actually. The 24 is very interesting. This is originally manually annotated as an endothelial cell. So we found that 24 is actually the lymphatic and the endothelial cells. They have those PDPM and the LYVE1 genes highly expressed, marking the lymphatic endothelial cells. So let me, this is, how the three kind of, real cell cluster methods doing. So you can see the gene class 3 here, they even cannot separate those abundant cell clusters. They not do good job at abundant, they not do good job at the real cell class either. So the gap cluster also don't do a good cluster job. They find the macrophage, they find the real cluster, but they didn't find the T cell and the T cell and the mast. The file as well, they find the macrophage, but they cannot separate, they cannot identify the mast and the T cells as the real cell types as well. So this is to show you how the optimal is, optimal data choose. Let me skip this. So next, the final thing I want to show you example in the spatial single cell clustering, spatial transcriptional clustering. This is not a single cell level. This is 10x vision. This dataset is commonly used as a benchmark to kind of evaluate all kinds of spatial clustering methods. So this is the H&E image. This is an anatomic reference from the animal mouse brain atlas. So people always, for those major clusters, most methods can identify very well. They generally will focus on this hippocampus structure here. The hippocampus structure, they have, two regions, the dorsal and the ventral. So in the dorsal, they have the dentigerous. They have CA1 and the CA3. For the ventral, they also have the dentigerous. They have the CA3 and the subiculum region. So actually, they have the 6 substructure there. So for this is, how our method identified those clusters. This is a kinase label. This is a kinase label at extremely high resolution. So let's focus on this sub-region, the hippocampus region here. So you can see here, we can clearly identify these 6 substructure very precisely match the H&E image here, So, but for the KN and the KN high, you can see they either can just identify 2 structure, or they can identify 4, but they cannot identify these 6. And they, for example, these 2, they kind of merged into the background, so they cannot identify those substructure out. So this is, I also compare the rare cell clustering methods. So we list the cluster, the 10 clusters with the lowest number of spots. So that means those are, we can see this is rare. We can treat it as rare. We can see each kind of rare cluster corresponded to a specific region and corresponded to a specific anatomic structure. That means they are biologically meaningful. But for those, real cells identified each methods, they are kind of scattered around. For example, gap cluster, they only identified this substructure. For the file, they identified this real substructure, but they also identified, this is actually a relatively common kind of structure there. So last thing I want to compare, actually it's quite amazing, So AKNO is only based on the transcriptome. Actually, this is a spatial transcriptome. That means you only have this transcriptome. You also have the spatial information. You also have the HE image information there. So there are a lot of methods developed trying to incorporate the spatial and the image information to improve the clustering performance. So some I listed the widely used like Bayesian space, graph ST, SPAGCM, ST-learn, DRSC. Some are kind of, those based on the graph convolution network like SPAGCM, those deep neural network methods. So also focus on this, structure here. You can see only the k-nearest label. Although the k-nearest label, adaptive k-nearest label only use the transcription information that can identify this substructure very well. Other methods, even they incorporate the spatial and image information, they not identify those substructure accurately. So to summarize, so the adaptive k-nearest label, because they choose K adaptively, for each cell or each spot, they can review the inherent cellular structure. They can identify abundant and rare cell types accurately and simultaneously. They applied on the spatial transcriptional datasets. It can, reflect the fine-grained anatomic structures. So the R packages are available in the GitHub. So it's very easy, if you use the traditional Serrat pipeline, you can just replace one line in your code. Then you can use the adaptive k-nearest label. Our paper is also available on the BioCAD. So I want to thank our team. This Jai is a postdoc in our group. She is the first author in this paper, and Dr. Yusha is another senior author in this paper. We also want to thank the grant support from multiple and collaborative grants. So I'd like to thank your attention. I'm more than happy to take any questions.